Today we are down here at Langston University to visit with Dr. Matand, who is doing some daylily research, and you've been working on this for quite a while. Tell us exactly what it is you're trying to achieve here.
Oh yeah, thank you very much for visiting us here. Definitely, because it's very important to share our secrets here with the world, especially the state of Oklahoma, because what we're doing is about Oklahoma.
Right.
So what we're doing here is what you see, we have several cultivars of daylily. And when you look around, you will see that where you have a flower, and then next to it, there is a dying flower at least.
Uh-huh.
So the reason is daylily simply bloom for 24 hours. Exactly.
Hence
After
the name, right? Daylily.
Exactly. The beauty of a day. So after attending and witnessing a great contribution of daylily association in Oklahoma here.
Mm-hmm. We got a lot of people working on daylily breeding here. Yeah.
Absolutely, absolutely. They do tremendous work, but they end up with the same result, 24 hours death.
Uh-huh.
So at Langston, we decided to go for a solution. And the solution is about trying to figure out how we can extend the life span of the bloom.
All right.
If you compare it to rose, it can go up to 10 days, but daylily is 24 hours.
And a lot of people might say, my daylily blooms a long time, but it's not the exact same flower, right?
Good point, because many people, what they don't know, they wake up in the backyard, they still see daylily flowers, but what they are seeing are flowers they did not see yesterday.
Right.
But it's the flowers for today, then tomorrow they will see flowers that will bloom tomorrow.
Right.
So that's basically a kind of a natural compensation.
Right, which is why a lot of people have bred for having more buds on the plant, but you're taking a different approach.
Definitely. So what we're doing here, we still do improvement of daylily, but not in a classical sense, because we are taking the approach of identifying the genes that are killing. Because the death of 24 that I'm talking about, they are basically inherited. They are genetic processes. So we have basically gone and extract all the genes, active genes, throughout the developmental stage of the flowering.
Okay.
Okay, and then what we did, we zoomed in the suspect section. That means transitioning from bloom to death.
All right.
That's basically the suspect section.
So in the lab, you've identified the genes that were responsible for flowering and the genes that are responsible for that flower to wilt and die.
Exactly, that is correct.
Okay.
Identified flowering from the early stage of budding to the death.
Right.
Yes, we captured all the genes.
Okay, so now that you know that section that you're targeting, what have you done with that?
Okay, what we have done with that is that we deliberately went into the suspect group of genes. So we aided with 1, 2, 3 letters at a time by we insert or we cut them out.
Okay.
Okay, so by doing this, although the gene might still produce the product, that product will no longer be the killing agent of the flower.
Okay.
So that's basically what we are hoping to end up with.
Okay, in my head, I'm visualizing flipping my breakers, trying to see which one controls the kitchen lights. Is that sort of essentially what we're looking at in a very simplified manner, right?
Actually, you are right. You are right.
Okay.
Because what we expect is that in the end, we will have a series of responses.
Okay.
Because what I just wanted to add is that those genes, after editing them, we put them back in the plant.
Okay, and how do you do that?
What we do is that we take the gene that has been already edited, and then we put it in a syringe. There is a special structure called the ovary. So we deliberately target that ovary because that's where all the ovules are, or female gametes are.
Which eventually will turn to seeds, right?
Exactly, exactly. When they fuse with the male, then they become an embryo, and that embryo will then grow into a new plant.
Okay.
So we target that one, we inject DNA that has been modified in there. And the reason we do that, we're not interfering at this point with any natural process of pollination.
Okay.
Because we let it take place so that it activates cell division in that ovary. And at the same time, when the cells are dividing using mitosis, they are at the same time incorporating also our DNA, we stick it into there.
Okay.
And then what will come through seeds, and then we'll screen which one basically has the marker indicating that it has the genes we put in there.
Okay.
And,
So that sounds like, though, a lot of your research, like, you're really busy right now. They're all in bloom. You're out there trying to get the genes in it, but it's very seasonal, right? Which can be problematic, I would imagine, for research.
What we did is we have a backup plan.
Oh, okay.
Instead of simply relying on a yearly season, we have a greenhouse, then we have the lab where we can use a tissue culture.
Okay.
Tissue culture so that we can work outside, even if it's snowing outside, we don't really mind. We just use the tissue and then work in the lab.
Okay.
And during the freezing or maybe the winter, we have the new plant will grow in the greenhouse, and then research continues.
So here you've got these, you're waiting for seeds to come, and then you actually have to grow those seeds out, and that, again, takes a process to get that plant to maturity to see if those flowers live longer than 24 hours, right?
That is correct.
How do you do the tissue culture process?
Or the tissue culture process we started with before actually reaching to the point of isolating genes. Because we anticipated this step, that at one point the field work might not work, then the backup plan should be tissue culture. So the tissue culture, we've already studied the whole plant of daylily. We know how each tissue reacts. And we cut it and then take the genes, put it inside the bacteria. That bacteria is called the agrobacterium.
Okay.
So, and we take that agrobacterium, when we cut the tissue, it is going to release some chemicals. So one of the chemicals basically is going to stimulate that bacteria to release or share its gene content with the plant.
Okay.
And that's what we are focusing on.
Okay.
At this time. Yeah.
So that bacteria is just a carrier of the genes, really, right?
That is correct.
Okay. And
that is correct.
Do you leave the bacteria? Does it ultimately do anything to that tissue?
Oh, okay, it can do, let's say if we don't kill the bacteria, after 3 days, then it will use tissue to survive.
Oh, okay.
And then it will overrun, and then definitely the tissue will die. So that's why after 3 days, then we transfer the material on the selection medium that also has one of the selection being targeted is the killing of the bacteria.
Oh, okay.
So we get rid of the bacteria.
Okay, and so then you're waiting for that bud that's got the genes inserted for it to actually start growing a stem and a new plant from that?
Yes, because the exchange will take place where there is a cut.
Okay.
So, and then also the signal we put in the medium is for the plant cell to pick up and then respond in forming the new plants. Okay. So among those new plants, we hopefully all of them will pick up the new genes, but very often it's not the case that we expect at least a few of them to pick up the new genes, and then that will be fine with us.
Well, it's fascinating research that you're doing, and I think a lot of times we focus on the flower and the color, but it's interesting that you've taken a different approach and are looking at the actual day length of the daylily. Thank you so much for sharing this research with us today.
Thank you very much for actually doing this to help people know what we are doing here at Langston for the people of Oklahoma.
We hope you enjoyed this video as part of our Oklahoma Gardening YouTube channel. You can also find even more videos on the OK Gardening Classics YouTube channel. And join us on social media for great gardening tips, photos, and discussion.